So I want to welcome the known faces of the wonderful talks that we have had earlier, but we also have two new faces whom I would like to briefly introduce, which is Tony Liu. He's currently the CEO at AbelZeta. He started at AbelZeta in 2013 as the chairman of the audit committee, and then became CFO and then CEO in 2016. And before that, well, he holds a Bachelor of Science from the Suzhou University in China, but has actually spent a lot of time in the US, in Seattle with Microsoft, doing very different things than he's doing now. And had leadership roles, different leadership roles, including the general manager of corporate strategy. And then in between, he also worked at the Alibaba Group, working both at the business to business, but also the business to customer operations as a corporate vice president. So welcome to the group. And then last but not least, I would like to introduce my colleague, Silvio Weber. Here at Miltenyi. Silvio is Scientific Director for Industrial Workflow Development. So he and his team are focused on developing closed systems on the Prodigy for our industrial partners, collaborators. And Silvio holds a diploma in biochemistry, as well as the PhD in the Universities of Bielefeld and Kiel. Welcome to you as well. So the idea or the topic of today's discussion is looking at the translation of CGT from academia to industry. This will largely, I could imagine, be focused on the process, but as we have such a distinct panel, I think we'll also touch on several other aspects, such as the clinical development, the operations. And maybe in order to kind of break the ice, I'll start with one question to Silvio, if I may, regarding what you see are the challenges of small biotechs or academia, which they may face in scaling up their manufacturing process in the cell and gene therapy space. And maybe before you go, so while I have a few questions for the panel, I think everyone is invited to chime in if there's topics that you want to share on the on the questions. I think I want to keep it as interactive as possible.
All right, thank you, Boris. Yeah, I assume the challenges are endless, but let me pick a few. I think lack of time, lack of resources, lack of access to critical raw materials, and a certain resistance, I think, to make changes to the process. So what we very often see is that early stage researchers came with their magic sauce or their very distinct process. But if you look a little bit later in development phases to have commercial viable processes, and this applies to both, not only manufacturing, but also analytics, well, this could be really difficult to transfer that. And so sometimes there should be some availability on this end to make certain changes towards more industrialized processes. So in this lack of time and lack of resources, they often they lead into certain lack of robustness later in processes, also in analytics. I think it's obvious, right? Speed is money. You have to move quickly. The market becomes more competitive. But sometimes it's really worth to take some time and to do really rigid research and a good basis of experiments to be sure that in the end you will really have robust assays and the process. So to bring that all together, these needs for robustness, but also the need to be quick, I think that's really a major hurdle to come up then with commercially viable processes in analytics.
Thanks, Silvio. Anybody else who would like to chime in?
I can start. I think I missed this, my major thing I wanted to say during my talk. Don't be afraid to make changes. actually the changes are important to be made on time rather than to live with suboptimal product afterwards for many years. It is challenging, but what Silvio said and my experience is do the parallel path. You keep doing what you're doing and at the same time try to find the best, the better option, I may say. And don't be afraid again to talk to the health authorities with the proposals, go to them for advices. They are very receptive and they help. They also understand that we need to link the clinical data and the testing as well at different stages, and they help out with that. But yes, I think it is very critical for all of us to understand that what we start with is a more or less proof of concept, and after that we move to something that can become a product for a long time. And we have lived that, and I think it's, I can speak from personal experience, it works.
Again, thanks for the invitation. I actually flew in from Shanghai, China. That's why these guys are very nice. Let me sit over here, not because of what we do, because from the furthest distance, I guess. For cell therapy, I think it is extremely important to be fully plugged in as a process is part of drug making, unlike for small molecule. And again, my background is physics. I have computer science, and I'm also a CPA, accountant. That tells you from my own experience, that's why I am the chairman, I'm CEO, I'm CFO, raise money, spend money, strategy all together. So talk about the making drug. One example I can give to you, echo your comments earlier, because for cell therapy, one change for reagents, that could make a big difference. One example we had was our two for the one of the drug we're making. And it was working beautifully, nicely, long duration response. But all of a sudden, there's a few patients, seems like not quite there, just missing a touch. Then we spent a bit more time, dug deep into it, realized the titration of the R2 was not there. We make changes, fix problem. But this is where now in our company that I organize from the manufacturing PD, clinical team, and analytics, all in together quickly. The minute you have any PK, PD, we take a look at, is it really performing as we talk about the curve, the USC, all that stuff, how useful the cells are, and the ratio of CD4, CD8, the potency, all these working together. This just tells you all this stuff, all deeply interconnected. So there's no such thing by accident. It's all work together as you do. So that's my own experiences. So yes, I wasn't a scientist, but as you spend 10 years with these people, you really have to not just strategically look at the big picture, targets get right, but also manufacturing process, every process in the way, you make sure you fully understood. So that's my 2 cents. Thank you.
Yeah, it's a great question. So I just wanted to share the personal experience. There's no doubt, right, for coming from academic center without like an industry partner, there is no way academic center can take the product from lab to the general people, right? To commercialize, we need it. But I think that it should be a two-way process. Like means both sides should listen to each other and learn from each other. One very good example that I want to share is with our CD19 CAR. When we make a product that we only proliferate for day 14, right? Because we all know NK cells has a problem for persistence. Like you cannot expand NK cells for a long time. So that was our product. Like if we had a chance, we would rather shorten the expansion period rather than prolong the expansion period, right? But that product was taken up by company, and company wanted to move forward with the product. But what company that now the research and things are gone out of the window, right? That company has their own set of standards. That means it has to make a profit. right? So for that, the company decided that, no, we don't want to do day 14 expansion because day 14 expansion gives us X number of cells. Instead, we do second stimulation and take the product to day 21 so that now we have 3 times more cells and 3 times more profit. But that doesn't happen, right? So that's the key information. Like, first of all, your product has to be effective, not more. So these are some nuances that we all need to like listen to each other and then learn from each other. Like academic has their own strength, industry has their own strength. When we can really truly make a good team who understands each other and respect each other, I think the sooner the product will be better.
Yeah, I want to add few comments on this, on the transition from academia, of course, where we work, and industry. There are other barriers, right? Including, you mentioned in your talk that you're having this great result in Hodgkin lymphoma, and people think that Hodgkin lymphoma is no longer an interesting disease because there are so many treatments there, right? And we are facing this problem in many conditions, because the more we start to become targeted and precise, the number of patients goes down, right? Because if you start to divide the number of lung cancer, breast cancer by molecular subtypes and going more and more expression of certain markers, the population and the fraction becomes smaller and smaller, right? So paradoxically, companies are losing a little bit interested in this approach, and they're sometimes going back to like broad approach. And one example that I had is about cancer vaccines, right? So Pfizer BioNTech, maybe we can hear directly from BioNTech, Moderna, right? They started with a personalized approach, right? Neoantigen approach, sequencing tumor, getting all the neoantigen, getting 30 of them, expressing in a vaccine, and then do one by one for each single patient a vaccine, right? This approach is kind of paying off. We see some positive result in pancreatic cancer and melanoma from Moderna and BioNTech. But now they are also starting to have a shared antigen approach, right? Something that is more applicable, more broad, easy to make, going against shared antigens, maybe less scientific compelling, but commercially probably more interesting for a company, right? In terms of cost and gains, right? So this, from academia, we found always a complicated barrier to overcome, right? Finding something interesting, and then the excitement of the company world is extremely low because numbers are not really there.
Thank you. Maybe as you mentioned, or as was mentioned before, the often for the cell and gene therapies, right, the process really impacts the cell and the function and activity. I was wondering whether on the clinical development side, whether you had experienced such transitions and changes in activity or also toxicity of products that may have come surprising or not.
Sure. I have a very dramatic story that I experienced about a week into my time at Juno. And so this is a story that probably a lot of you already know. And this was a CAR T-cell therapy we were developing for ALL, adult ALL, called JCAR15. And I was called by my boss at 6 a.m. asking me to come in to help out because we had a patient who had severe cerebral edema. And so this was a CAR T-cell that was brought in from academics. And I think we've all learned a lot about looking for, transferring technology from one manufacturer to another. And I think what we discovered was that we had a CAR T-cell that was much more active, manufactured in our manufacturing suite than others. There are a lot of other things that we found, and I think you've probably read, but certainly that was a hard lesson to learn. So we had several patients who they experienced cerebral edema, which ultimately was fatal, and that project was discontinued. But again, a lesson learned. I think now we know how to analyze the cells to look for the differences and make sure that there is equivalence. And I think things that have already been pointed out, small changes different reagents, different containers can make a big difference in the happiness of the cells or the activity of the cells. Anyone else?
Maybe had a follow-up question on that one.
Yeah.
If I may, so you mentioned you now know what assays to use, what to look at. One of the big topics in the field is the potency assay and what it really tells us. So I was wondering whether that may be the same assay that you were looking at, or were those exploratory assays that you had to add to really get to the crux of the therapy?
That is a good question. I think, we had some, I don't know the details of the potency assays at the time, but what we did see, a very strong difference between the expansion. We had very rapid expansion of those CAR T cells. So these are very gross differences compared to in the academic manufacturing process. We also had in particular, rapid expansion of CD8 CAR T cells, and that was associated with those patients who experienced that toxicity. Not all patients experienced that toxicity. Certainly, quite a number were treated without that, but when it did happen, it was dramatic, needless to say.
Thank you. Any comments to this topic?
And maybe just one thought to that, what you said. So this dilemma that we face, that we have maybe less and less patients for personalized or precision medicine. I think the example from Stefan Miltenyi in the beginning shows nicely that maybe a different way of thinking, that you don't look for the best in class, but rather you have one very nicely working therapy and you apply it to a different disease setting, could be a nice option. So maybe there are also other diseases where a certain asset will work in. So let's say you have a 2019, can work in autoimmune disease, in oncology, and maybe we just have to look a little bit more to the molecular signatures and trying to understand them and how we could apply the treatments that are already out there. So like repurposing, I maybe can call it. So this could be a nice way to overcome this issue.
So today we have active programs, work with J&J for CD2019 BiCAR. We out-licensed to J&J last year. We also have 3 active programs with AstraZeneca on solid tumor. I think the process of, through BD process, working with them, you realize whatever you design today or tomorrow, you need to keep in mind that potentially or eventually will go commercialization. So things like make sure automate it as much as possible. That's one. From that perspective, today we look at, I'm a big fan of Miltenyi system. I was here in 2018, by the way, and in this very room, talk about collaboration. I was often joked about I need to collect my commissions from Miltenyi because I've been a big fan of continued sales program for them. But the system continue today, product is about 90% automated, but still have 10%. So we look at robotic arm. We look at also robots, cleaning room, etc. Because the beauty of the systematically have a process in place in the future when you work with the partner, commercial partner. There's many reasons to do that because one, from academic to commercial, simply you don't have that much money or resource or expertise. So you want to keep in mind, as you hand off, if you can automate it, ideally you can give them, say, here's a disk, here's the parameters that will help you from the, today, tech transfer. Today, more like you do it and you copy, somebody will stand behind you, make sure you do the right thing. But, Mark maybe do it better than Tony or Leo, but that's not the way long term. You want to automate it because cell therapy, one, efficacy is so good, but the cost is so high that we do anything we can automate it. So that's where, from our experiences, that we need to continue to make sure from timeline takes, not only academic from the theoretical approach versus commercial, you want to make sure automated, systematic, and so that reduce cost. That's the only way eventually make sure industry can take off. And so whenever we do, we want to keep those factors in mind, less human. So my ideal the process would be that in EO2 that we have the remote control outside of the clean room. I talked to Stefan this morning, this afternoon. He said, someday we don't need a clean room. That would be ideal. Just use your system. I said, well, maybe how about in vivo expansion without even going to machine? Because you have to constantly think about innovation. For today, I know audience have a lot from academic background. So my point is that from the academic, as you continue experiment different things, not only make sure the target itself, but also manufacturing process makes it repeatable, replicable, and also transfer in a way that's more efficient. Thank you.
I can add on that from the academic standpoint. In academic lab, we don't have those robotics, or at least in the lab. But at least we are in the forefront. Why? Because we are thinking about it constantly. So at least when we are using the things in the lab, like for NK expansion, we try to use systems or things that are at least a GMP validated product, right? Because if you don't do that, you spend another year just by changing the media or even isolation kit or something for a year or two just to make them GMP compliant. So at least in an academic setting, what we are constantly thinking is like whatever experiments we do, we try to make sure that some of the experiments we can even use it as an engineering run for our IND submission, or if not, at least try to use those reagents, which are GMP validated reagents, so that later on when you tech transfer to the GMP, you don't have to worry about all these things because you can immediately transfer it to the GMP. So these are the exact, and then then from there it's a different ballgame. I haven't seen that, but at least in a smaller things, we are also constantly thinking about it, how to make it faster.
Yeah, I can add something to this comment. For our ALCAR trial, right, when we first produced the virus, and then we went discussing with the CMCF facility at Dana-Farber how to do the validation run for the FDA and then eventually do the clinical trial. And of course, they had the option to do manually, right, and produce the CAR-T manually or via the Prodigy, right? The problem at the time that for some reason, at least in the facility in Boston, they had been using the Prodigy only for lentiviral product, not for retroviral product, right? So they did not have a protocol. And so we spent a couple of months discussing whether we knew that manually would have been worked quite well because that was the way we were doing in the lab, or whether spend sometimes adjust the protocol to the Prodigy, use retrovirus on the Prodigy, although exactly they did not have experience. And of course, we contacted Miltenyi, all the technical office, and they help a lot. They had example at the Memorial Sloan Kettering, people using the Prodigy for retrovirus transduction, and they gave us a poster. They gave us, there are differences like using vectorfusing instead of other reagents specific for retrovirus, right? And we were pushed by the leadership of the cell factory to go via the Prodigy exactly for this reason, right? They told us, look, if you do a phase 1 trial all manually, let's assume it's going to work. It will take maybe a long time, many, many months, if not years, to transfer from a manual process to an automatic process, right? Better do now, spend some time now, and then eventually turn out to be a good solution because things work from the first day, so we were kind of lucky, or the virus was good, etc. But we had this discussion for several weeks whether the was better to do an early move in adjusting the automatic process, or whether delay it later, right? And I'm glad we decided to go on the Prodigy, and now it's relatively automated, of course, in an academia setting.
Thank you. That may resonate maybe a little bit with your experience, right? Transferring a process cookie from the UCL to the commercial facility. I was wondering whether you had you touched on it in your talk a little bit, mainly on the later stages, but I'm curious on the early stages. Are there any lessons learned, any transfer, or any early changes that may have made the transition smoother?
Yes, there are many. Huh? Do you hear me? Okay. Yes, I learn every day, and I'm still, I don't know, not there yet. What I want to add to, and I'll answer your question, but what I want to add about the product and probably you got lucky, yes. I mean, Prodigy is a good system, no doubt about that, but probably you got lucky as well. But there is no reason not to move forward with the Prodigy and to still to go for the first phase with what you have confirmed to work. The thing is, this process characterization, understanding the product is a key, and knowing what we are looking for is extremely essential, right? The people sometimes, oh, how many cells we have, how many transduced cells we have, and that's it. But how the kinetics goes, what is the phenotype of the cells, how they change if you do this and that, these things needs to be done very early, and this is where the talent is important, right, for us. The process A versus process C. You saw this beautiful thing I had over there. Process A in UCL and process C, right, Prodigy, almost fully automated, semi-automated, but we're going there, right, Silvio? Right? Okay, yeah, I have huge faith in you. And in the beginning, when all the things started, with the process A, when the whole cells are not selected, you have the crude PBMCs and you add the vector. Of course, it is going to be a different thing. Of course, it is going to, when you do the transduction, efficiency is going to be lower. But this should not stop you to say, no, those processes actually are comparable, and they are comparable because of this and that and that. So when do the comparability as often as possible, pick your parameters to compare and analyze them properly based on the science, not on the feeling, right? And yeah, I've heard this a lot. Oh, I feel, yeah, good, me too. but you know, it's probably not a good joke. But I mean, I'm hearing this a lot, yeah, the comparability is a key. Find somebody in your teams that actually is going to become the king or the queen of developing these comparability protocols and do it as early as possible and do the characterization. Doesn't matter. You change your reagent for your asset. I mean, characterize it. Do the comparability between these two reagents, because sometimes the analytic comes, oh, you have totally different expression here. No, I didn't change anything in the process. No, you did. I have this. No. But did you, oh, we bought something new, right? And this is a concept in the early stage that is not familiar. People are not familiar with that, right? They change the lots, they change the vendors because it is cheaper here than there. And you're right. I mean, this causes months of delays. But yeah, this, I have learned a lot, and the things could have been done earlier. Yes, but it is also very powerful to understand the things later, because a lot of patients actually have been treated at early stage and they're still in the remission stage. So we have to confirm that actually our product was the same. It doesn't matter that we changed the process, right? I think it's an art. It's not like you guys are much smarter, right, with these medical doctors. I adore you. But I mean, this is also some kind of interesting field in the development. I love it a lot, actually. Yeah.
Cookie, I wonder how difficult it is to control things like, I mean, the starting material, serum, HSA. Yeah, I mean, this is something you have difficulties to control. So what is your take on how to make sure that nevertheless your process is controllable and robust?
Yeah, the human AB serum, right? I mean, this is $1,000 question. The good news, there is a good news and bad news, right? The control of material needs to be controlled from the centers where actually the donors come before the process comes. And when the supply chain goes and when the procurement goes, I encourage the process people in your organizations actually to go to and see how the process is, how they made this product, and ensure that any change they have is communicated to you, right? I mean, to any organization. Because it is important, if something changes and you don't see the reason, you don't see changes, fine. But if they have made the changes and you don't know this is, and you start seeing differences, you start going towards process, people, analytics, but sometimes it is the raw material. I think Miltenyi specifically is in the business where you provide a lot of our critical reagents, and we're in constant communication with you about the changes, so what is happening, and we're also doing this to kind of to collaborate to know what is going on. And I think it's the collaboration is my answer to your question, but at the same time look for something that is going to substitute slowly but surely. We all have to do this continuous development and continuous innovation. For the future products. What is going to happen? Are we going to get some growth factors? The hubs is good, we put them in excess. They have the secret sauce, right? But some people say, if you gamma irradiate that, what is going to happen? Is it good? I mean, yeah, I think that the answer is continue to look for something that is more reliable and more defined, like the serum defined, right, chemically defined serum, which hopefully we can use in near future for the CAR-Ts. But yeah, it's tough, but it is possible. It is under control, right? It should be under control.
No, I was going to make a comment about the earlier further to we're talking about the build a new core organization where the head of the PD, head of AD, head of manufacturing process and quality, almost we have a war room type of team. Every patient, particularly for early on, that 10 pages, I don't know exactly, even happens any changes, supply chain, that because anything particular could impact overall downstream. Typically in the old days, you want to have the clinical team. You do not want to know what's going on over there in favor of losing some trade secrets IP, but over here, that I do things by trust. And you have the rules, process, so we talk to each other all the time, relay. Here's what manufacturing is. Look at the potency, and look at the clinical development team side results, and look at the expansion PK, whether day 14, or what happened on day 7, how day 21. So it's all related. But for Miltenyi, I would say development side, it would be, I talked about last time when I was here, it would be ideal if you can download all the digital information. Yes, process reagents could be changing from the raw material to different patients. However, if you can download the digital information, that in my mind, someday, from 10 patients to 100 to 1,000, you should be able to build the chart to, ideally you can predict even before patients got treated, say this patient got 85 or 90% chance of being, successful. That's what I think, not from the academic to early to manufacturing process, developers to industry, really to work together. And I think that, our product, we out-licensed J&J. Certainly, yes, we're competing with you over here. We saw 85% complete response for the last line for lymphoma, AbelZeta. That's published at the last year's ASH. Now we look at every single patient very closely to a point where any minor changes, we know exactly, we can tell what happened. So I think it's key is constantly can break the barrier. Team talk to each other. But once you finalize, stay as is, then you don't make changes.
Thank you. Maybe coming back to the clinical experience with changes in the process, is there anything that you recommend or have seen in the past that changes in the process, although well controlled, may have impacted the clinical outcome?
Yeah, so I think we all would love to start a clinical trial, never change the process, go ahead and commercialize it. That's the ideal, but that never happens. I think a really nice story is the story of Breyanzi. So when we started development with Breyanzi, we called it JCAR-17, and it was initially being studied at Seattle Children's in pediatrics, and so the initial process was not scalable to adults, so we had to quickly change the process. So we quickly went from version 1 to version 2. Then throughout the process of development in lymphoma, we ultimately had 4 total process changes, but just as you pointed out, each process changed, we demonstrated clinical equivalence. So we were able to ultimately, we had a seamless phase 1 design where we ultimately submitted that data for approval, and we now know this is approved for lymphoma. It's called Breyanzi. But in other programs, so for example, CLL, we did make the decision to delay starting that trial until we were more certain with our process. And in fact, the CLL program was started with version 4. So that trial was done in sort of the ideal way where you don't have process changes. So that's one example. I'm currently living through another example at BioNTech where the trial that I shared with you was started with a manual process, and then a decision was made to move into the automated process with the Prodigy. When they performed the testing to try to demonstrate equivalence, they noticed that there was actually quite a lot of difference between the cells. The cells were much more active, and so it was, the decision was made that the phase 1 trial should be repeated, basically. And they even went down a dose level lower than the original dose level 1, just to be ultra cautious. And now, things are going well with this process, and I think maybe hopefully we are in a process with minor tweaks that would be commercializable. But the reality is, I think going through an entire clinical development program without process changes is probably not realistic, although ideal.
Can I add something here? Because we very much depend on our clinical colleagues to define the dose. And usually the dose starts, low. In some trials there is dose escalation, right? But when the process is designed, it is designed pretty much for lower dose, I may say. So when it ends up the dose to be high, the process needs to change, right? What you say. So I always try to design the process from backwards and assuming that this is the worst case scenario. If you have to have 500 million CAR-Ts, for example, or 1 billion CAR-Ts, you pick the number and you design your process backwards depending on the dose, because it is going to be very difficult afterwards to do that. And it is the same thing with the scale up with the other products, right? It doesn't matter if you need the dose, you can put extra step, but design it for higher dose so you can have a larger, broader reach later, right?
So for us, being like a allogenic product, it's easier for us, right? Because we define a dose, we freeze it before we even treat the patient. So, yeah, you are correct. While we define the dose, for clinical trial, it's easy because it's defined how many doses for how many patients. Like this is the dose for a patient, this many patients. So we have a statistician who calculate that. So we freeze multiple vials. So some vials will be with lower doses, and some vials will be with the higher doses. So later on, when we had to go back or go up, either we thaw 2 vials. So we have a condition, as I told you, our product 1, we froze too many vials with a lower dose. And then now we have a patient with the dose level 5, then now we had to thaw 100 vials instead of 1 vial, which is not, if we have to do it, we have to do it. But luckily we have another product which had little higher dose, so we can do lower vials, right? So that's a drawback for this allogenic product, but eventually that's the thing. That's something that I think when we really got into the phase 2 or in commercialization, we really need to go down, like it has to be really different than what we did in the clinical trial, right? There, I think they will be approved for one particular dose rather than like this variation in dose in clinical trials. So I don't know. I think you guys are expert on that. We haven't thought about that there yet. But yeah, definitely we think about dose. We freeze multiple vials with different doses so that we can carry our clinical trials for different amounts. So, and to tell you about this process changes, right? We are doing process changes constantly. One of the main hurdle was COVID, right? When COVID happened, we were using this media from Cellgenics, which is from Germany actually. They were making this NK media. So when COVID happened, they completely shut down the media production. And then the factory was repurposed for COVID vaccines or something. And all of a sudden, we have all these patients, and there is no media that we can use. So we had to panic all of a sudden, and we had to go through like this process development where we had some media, and we compared them with other medias that are available, which were the other alternatives. So we had to spend like day and night like doing these experiments and validating that in this different media, these NK cells are behaving same, and then we had to go contact the FDA and change the whole protocol. So that's a small thing, like a big thing. And every now and then we'll be changing some cytokines or some way how we expand NK cells, because since we have a luxury at this point while we're doing clinical trial to make our product more potent, we quickly know whether this current regimen is working or not. So maybe we are already constantly in the lab thinking about how we can make better. So if we find some alternative sources, how we can make it better, we can quickly do these comparison assays. Of course, we had to do all these in vitro, in vivo assays, and then make this file and send it to FDA and say that, okay, this changes. Sometimes there is a case where FDA asks us to go one dose, like as you said, like a couple of doses lower to start again. And in some cases, they were okay and say, okay, go ahead and please make those changes. So.
Thank you. Maybe coming back to the kind of early topic of academia transition into industry, Roberto, I wonder whether you have any experience with your development of CAR-Ts and the potential also of academic institutes maybe to register and have a drug, or is there dependency on industry?
Yeah, so I think the way we are set now is we depend on industry, right, for the next step. Actually, even when we submit grant to like some foundation or some funding agency, they always ask, okay, if you do the trial, you get some positive result. What's next? What are your plans, right? I mean, because they know that the academia reach a certain level, and then they tend not to think to the next step of expanding the production and commercializing. And so now the recent grants tend to ask this question, right? Well, if there are plans in place. So that's a tricky question. I think for the situation as it is now, the process is so expensive and so complex that, as mentioned before, cannot be done in academia, right? But in the future, that could be not so far away. I mean, we need to think that maybe this automated process has to get out from industry and go inside hospitals, right? I mean, the production, if we reach a point in which is really not only 90%, but maybe 99.9% fully automated, the system, they should start to decentralize and go in periphery, right? Because the way it is organized now, it's a lot of complexity and extra cost, right? Having few European centers. I mean, for example, in Italy, right, we are treating patients with CD19 CAR with KITE, right? And the manufacturer is in the Netherlands, so we need to ship cells from Italy to the Netherlands, wait and get them back, right? Is extremely complicated and expensive process. So we hope that in the academia there will be one day the chance to have beautiful instruments, like very small, that they fit on a bench, And with minimal manipulation, you can generate your product very fast in a few days, and directly injecting patient locally, right? So I think that's something that maybe Miltenyi could really help in reaching this point.
I can add something since I'm from academia. I think I agree with you. It might be doable, but I don't think it will be a right thing to do, right? Because if he has to come from the academic center, I think nowadays, like MD Anderson is thinking about that, they have this tech transfer office, commercialization office and everything. But we need to think about it, like in academic setting, okay, you can make the product, but making product is not enough in today's day, right? You had to first commercialize it. That's a different ballgame because there are so many products which are FDA approved, but they are not viable just because they can commercialize or market it. So there are different aspects which I don't think in near future academic center won't be able to do that. Just by making product is not enough. Just because you have other puzzles you have to be coming into the same in a place in order to make this product like viable or be profitable, right? So I think until and unless these academic centers are very forward thinking and they distinguish these different phases in different way. Like once the product is viable, they transfer it from the lab like to a different center where lab PI doesn't have to do with anything. Like there is a different separate unit who handles like production, and there is a different unit where they handle marketing and making it a viable product or commercialize it. I think at least I see it's not possible in near future. So I think we still need to rely on industries in order to commercialize the product. But nowadays the good thing is all universities are being aware of it, and every university are trying to, or academic center or research centers are being aware of it and trying to set up a tech transfer office or commercialization office, or trying to invest some on it. Especially now people are seeing that these academic centers are getting a lot of grants and money from intellectual properties by giving their findings as intellectual properties and stuff like that. So hopefully in near future it will be, but it might take some time.
Just a quick comment. Today, I look at, we're close to the end of the hour. I think the Miltenyi bring academics and industry and all together is a great start, because making a CAR-T drug is a long journey. It requires a lot of, we talk about money, technology, resource, process. Perhaps, let's say Miltenyi, from academic perspective, for scientists to find a good target is already hard enough. But then think about money, process. Industry probably will go raise money, and half my job is raise money, right? But for academics, may not have the luxury. Rely on grant may not be enough. Maybe Miltenyi can provide selectively yearly a few academics. I'll give you machine for free, but certainly you select targets. You like the target. Because the beauty of that is once you use it, make the next layer, then people will use your system for bigger business, and it's not for free, right? Or even lease it. That way you help get rid of the initial capital need. Couple $100,000 is not cheap, but if you lease it, just like buying a house, you don't have to, right? So those are ideas. But for us, digital information that helps. And also best practice sharing. These guys can help because ultimately the grassroots will go up. A few get somewhere, and then to the final, hold it well. Some ideas.
Yeah, I think there's several aspects where we're trying to enable hospitals and academia. I think Stefan mentioned one of the commercially available vectors earlier. So I think there's different avenues. I would like to open up to the audience as well if there's any burning questions or questions that were found in the chat. Otherwise, maybe in the meantime, I have one more question for you, Tony, with regard to how you see the role of smaller biotechs in the cell and gene therapy space with the bigger competing pharma. Where do you think there are opportunities for smaller companies, and what kind of business?
Sure. I think that we are in the, I would say, between academics and large commercial company. And our company is about 400 people today. Annual cash burn about 100 million. It's sizable. So where we think that for me to become a large global pharma, it's going to take another 7, 10 years. But in between, we serve the role. We have quite a few assets, no longer the early stage in terms of try different things. So selectively, we have 7, 8 programs going. So our role is extremely important for a company like Miltenyi, in that anything we have, for instance, our CD2019 program, we out-licensed J&J. Our process, and but now as is, didn't make any changes. Now going to the next phase. Because for Miltenyi, also means once you commercialize, means a lot of machines, a lot of systems. But for us, is that through our licensing, now that program, I don't need to spend money anymore because it requires hundreds of millions, if not billions, to go to the next phase. Then you have other programs. So, but I also need to make sure we clearly define our role, where no longer at the small program try, but more sort of half in between, but not commercial enough yet. So we have a few other programs going. So it's important that we want to make sure, because we have now with J&J, early days we also work with Novartis, now AstraZeneca, 3 programs. Extremely important, anything we do, impact downstream. Not only make sure the quality of the, talk about the quality of the program itself, but also manufacturing process. We talk a lot about changes. Anything we do that has downstream impact, because potentially that impact the time to the commercialization, BLA. Every change, it matters. So this is where I talk about automation. I spend quite a bit of time in the automated process, so that in the future, ideally, you would have a handbook. You give them exact digital parameters. Here's what we did. Here's what we do. Here's what needed to be done. At the end of the day, it's about the marathon. All of us work together as an industry to tackle those mathematical diseases. In a sense, you don't try to boil the ocean all at once. You know your role, know where your place, but set limit where it's like go to a casino where you gamble. You don't gamble too big, but not too small, and then go beyond your comfort zone limit. So that's where, as a CEO, I constantly evaluate programs. Some programs require a lot of money. I may not be there today, but some of them try to place bets at different places. It is doing drug, I'm sure, too, have a lot of experiences that you have one step wrong, you cannot repeat, you cannot go back. In the Chinese saying, 过了这个村就没这个店, meaning once you pass this village, the same restaurant may not be there anymore for you. So it's extremely important. Thank you.
Thank you. Yep.
Sunil, I would like to take up with your comment, Benjamin Lüscher, Processing Facility Director at Labor for Cellular Therapies, Inselspital in Bern. I completely disagree with you. Because we are currently building up a facility to generate our own CAR T cells. And we may not go to marketing and stuff like this, but think about the concept of hospital exemptions. I mean, there are people which are excluded from clinical trials where we as hospital can start to produce CAR T cells for people which have no other which are out of therapies. And then, of course, the point of care concept from Miltenyi, which is quite interesting for us also.
Thank you. So you did not agree with the production, like academic center can be able to produce. That's what you're saying, right? Same, yeah. No, don't get me wrong. We are trying to do CD70 trial, phase 3 by ourselves, if possible. What I'm saying is, even FDA approved your product, okay? Then there is still a hurdle to market that product. Doesn't that, once your FDA approve your product, like it's not like next day the doctor will prescribe it or like the patient will get it.
But if you work in a hospital, you don't have to market it's not a big production, it's a smaller scale.
Yeah, sorry, I don't have expert like mind to tackle that. Maybe you can.
I'll take a cheap shot. No, I before I came here, I made my mind is no, I'm not going to argue with Stefan or Toon. The point of care, to me, has a lot of long-term vision goal. It's a bold one. That from the industry side, that you know, particularly when you come from China background, you see a lot of poor people need some access. So from that side, you need to. However, it's a long marathon ecosystem. If you don't, biotech, if you cannot make money, you cannot feed the system. You cannot get legs there because you're not here doing charity work. And there's days when patients come to me that you cannot, I have tears and trying to find ways to help them. But for this to work, we have a couple of companies in China, the partner for Juno, WuXi Juno, and Fosun-Kite. The product already approved, but try to commercialize it. It's very difficult because the license, the insurance. But go back to the question of medical center. But for medical insurance company to pay for it, you have to have basis. You cannot say, I can do it, here's my number. What's your basis for that number? Is it approved by the regulators? What's the cost of it? Who's responsible for the quality? How about this hospital versus that hospital? Who's going to say number 1 versus number 18, then number 98, and number 980? These different hospitals have different standards. How is going to insurance pay for it? Who is going to pick up, right? So there's a lot of things. If there's no standards, I know this is a long, long debate, but that's why I say I'll take one. I debated. I want to. That's why we out-license J&J outside of China, within China. Let me figure out what's the best way to help these patients. I don't want to make a lot of money, but I need to sustain the company so that the company can survive. That's the only way long-term we can be here. If there's no money going, whether academics, even for academics for the hospital, when they manufacture, you need to have the quality people. You have the products. They cannot possibly have a lot of IPs, different vectors, right? There's a lot of, it's complicated, but that's why I commend Toon has done a lot of work. I like the work Miltenyi is doing. But it's a long, long journey, lot of work. So I wish you all the luck. I really commend. I'm happy for you. I champion for you. But we're not there yet today. So that's, yeah.
And you need to think about the line of therapy. I don't know about, like in Europe it might be different, but in US, like it has to be in like which line of therapy. Like our NK cell might be in 10th line of therapy. So there's no way doctor gonna prescribe that, right? If you have like 9 different. So there, that's my. You can completely disagree with me, but my small brain can think that we definitely need, even though we run our phase 3 trial and we get FDA approved because we can making products, but make it a viable product which makes profit and which doctors prescribes it, and we can find it in the community hospital. We definitely need a partner, and I don't say it has to be academics like in some companies. Maybe MD Anderson or some academic center can make an entity which are specialized for commercialization and then taking it to the in a broader public. That's what I'm saying. And I think like MD Anderson, one of the biggest center for, like we run like more than 1,100 clinical trials, and even MD Anderson doesn't have it. I think Dana-Farber same. They don't have a commercialization entity. I don't think any other hospitals have that yet. So that's all my point. So for the long, we can do a sprint, but that last, if we run a marathon, the last steps, which is necessary to make money, you need some kind of help. That's all I'm saying.
I think we have to end when it gets interesting and spicy, but feel free to continue. I want to thank you all. I think it was a really interesting discussion with the different views. I hope for the near future we'll have different paths to bring cell and gene therapies to the patients. So yeah, will be remain exciting. So thanks so much for joining.